Hello, Physiology 142 students. Welcome back to the rest of our lecture on the digestive system. Now remember last time we were talking about the GI organs, which included the stomach, the intestines, and so forth, and those are the hollow tubes through which food passed as it's being digested. But we also have accessory organs that are not part of that hollow tube, and they consist of things like the pancreas and also the liver. Now last time we said that the pancreas has all these digestive functions as well as endocrine functions. It produces trypsin and chymotrypsin, which help to break down proteins, but it also produces bicarbonate, which helps to neutralize stomach acid. Now let's go on and take a look at the liver and gallbladder. The liver is a very large organ, probably the largest and heaviest internal organ, and it's located right underneath the diaphragm. The liver is divided into lobes, and each lobe is divided into lobules, and these lobules are composed of cells called hepatocytes. Remember that hepato means liver, and cyte just means cell. So here you can see one of those liver lobules, and the liver lobule is supplied by plumbing of the portal triad. The triad here has three different parts: the hepatic artery, a hepatic portal vein, and a bile duct. And we'll talk about what those do in the next slide. But what you'll see is that normally these lobules are sort of octagonal in shape, and they're organized around a central vein. Remember, the liver has a lot of different functions, not just for digestion. It helps to bioconvert one thing to another. It helps to detoxify some compounds in the body. So a lot of filtration is going on here. Okay, let's take a look at that hepatic or that portal triad that we talked about. So first of all, we have a hepatic artery, which is carrying oxygen-rich, oxygen-rich blood from the heart in order to supply the liver for its own metabolic functions. It also has something called the hepatic portal vein. Now, hepatic portal vein, which is over on your right-hand side, is an area that is basically bringing nutrient-rich blood from the intestines. As that food is absorbed, it goes from the intestines directly to the liver, and we call it a portal vein because it links two capillary beds. And finally, we have something called the bile duct, and of course the bile duct is the duct through which bile travels to the gallbladder for storage. Bile is actually made in the liver itself, and bile is a byproduct of red blood cell recycling. Okay, so the gallbladder itself doesn't really secrete bile. It doesn't make bile, at least. Okay, it secretes it, it stores it, it concentrates it, but technically the bile itself is made in the liver and stored in the gallbladder. Now, this bile consists partially of cholesterol, something called lecithin, various pigments that come from the breakdown of red blood cells, and of course bile salts. Now, bile itself is not an enzyme. It doesn't actually break things down in the enzymatic sense, but it does help to emulsify lipids. And so it acts basically like a soap. If you take a soap or a greasy dish and put it in the sink, and you see all that grease, congealed on top of it on the water, and it's just making a big film, and when you add a little bit of dish soap to it, it helps to emulsify or break that fat down into smaller globules, and that here is the job of bile. It helps to emulsify the fats in our diet so that they can more easily be broken down by the fat digesting enzymes that we have in the body. Okay, one of the questions here on the slide is, what is stercobilin? Remember, the different colors that we have in the body are often derived from red blood cell recycling. Red blood cells are red because of hemoglobin. And then we said that green here is biliverdin, which is a metabolic byproduct of that red blood cell recycling. Now, eventually, when the bile moves into the large intestine and small intestine in the opposite order, it is going to be converted into something called stercobilin. Stercobilin is brown, and this is the reason why poo or feces looks brown some of the times, and it's because of this metabolism of what was initially bile but now has become stercobilin. Okay, let's take a look at some of the liver functions. First of all, the liver is important for carbohydrate metabolism and also blood sugar and glucose balance. When you eat a meal, you absorb a lot of glucose from the simple sugars in your diet, and you can only keep so much in your bloodstream. So any excess sugar is going to be converted into glycogen, which is a storage polysaccharide, or it can even be converted into triglycerides. Both of these can be stored in the liver, glycogen especially. The other thing that the liver does is store some triglycerides, and we just talked about that. The liver can actually become quite fatty under some metabolic conditions. If you've ever heard of foie gras, that's basically a goose that we fill full of food so much that its liver fattens up, and apparently it's really great to eat. I've never had it before. It sounds kind of cruel, but we do store some lipids in our liver. And we also have the important function of deaminating amino acids. Remember that amino acids come from protein breakdown. So as we eat proteins in the diet, the byproducts of those proteins can become toxic unless they're detoxified in the liver. And so that's one of the jobs of the liver. Now, interestingly enough, if we have people with compromised liver function and they eat too much protein, they can actually get encephalopathy. They can become confused and just out of sorts because they're having so much urea and all these other metabolic byproducts circulating in their blood, reaching their brain, that it affects their mentation. The other thing that liver does, unrelated to digestion, is to synthesize many of our plasma proteins. And those are things like fibrin and fibrinogen. Those help with blood clotting. Our albumin helped with absorption of or reabsorption of fluids during the whole reabsorption and filtration thing going on in the capillary beds. And of course, we have other plasma proteins as well, such as well, let me think, our gamma globulins and things like that. All right, the liver does help to detoxify drugs. When you give somebody a drug like prednisone, it gets metabolized into prednisolone, and that determines that metabolism, how effective the drug is, how potent it is, and how long it lasts in the body. And so if people have compromised liver function, you're oftentimes going to use a lot less of that drug, because if the liver is where that drug is detoxified, it's going to take it a lot longer. And so we need to be aware of somebody with impaired liver function if we're trying to treat them with a normal course of drugs. And of course, the liver, like the spleen, is a site for removal and destruction of old red blood cells. Remember, that was the primary job of the spleen, but the liver does it as well. And especially if the spleen is removed, the liver can take over that function entirely. Also, the liver is important for storage of fat-soluble vitamins. These are A, D, E, and K. And also it helps us to excrete excess bilirubin, right? That goes out in the bile and eventually is converted into stercobilin. And finally, the liver is important for synthesis of the bile salts that end up in the bile themselves. Remember, bile is useful to emulsify the fats that are in their diets, so that our fat-digesting enzymes, the lipases, which are secreted in the pancreas, can actually break the fats down into monoglycerides and free fatty acids. Oh, last but not least, the liver also helps us to activate vitamin D. Okay, one of the conditions you probably heard of is something called jaundice, and jaundice or icterus is a yellow coloration of the skin, the mucous membranes, the sclera of the eyes, and oftentimes it's caused by impairment of the liver. We can see this in very young neonate babies. That's just a matter of course as their liver begins to accept the function of normal body function. So it might take a while. They might be initially a little jaundiced. But if we see it in adults, it's usually a sign of some sort of liver disease. Either it can be very acute, like in hepatitis, or chronic. It can be due to cirrhosis and alcoholism and things like that. So if we start to see this accumulation of yellow, that means that this what is the name of bilirubin, excuse me, that bilirubin is building up, and bilirubin is yellow. And if the liver can't excrete that bilirubin in bile, we'll start to see this buildup of bilirubin within the sclera and within the skin as well. Okay, now we're moving on to the last third of the lecture, and this last third of the lecture takes a look at what's happening inside the small and large intestines. Okay, the goals of this lecture are to identify the external anatomy of the small and large intestines, describe which features contribute to the large absorptive area of the small intestine, discuss the chemical and mechanical digestive processes that happen in the digestive system, and describe the ways in which different types of molecules are absorbed across intestinal mucosa. And finally, we're going to talk about the digestive, absorptive, and other functions of the large intestine. Okay, let's start out with the small intestine. The main function of the small intestine, of course, is absorption. There is a lot of digestion going on there. Don't let me tell you there's not, but this is the single organ in the body that its primary job is absorption of all those great nutrients that we've been able to extract from our food. The small intestine has a large surface area, and that's important because the greater the surface area we have, the better we are at diffusion and osmosis and absorption. Now, 4 factors contribute to this large surface area, and they include its length. It's about 10 feet long, which is pretty darn long. It also has circular folds in there that you can see up in your top right, and those help to increase surface area as well. And then if we look on those folds at a microscopic level, we'll see little finger-like projections, which are called villi. And villi, again, finger-like projections, more surface area around those fingers, help to increase the surface area for absorption. And finally, if we look in the microscopic level, at the cells that make up those actual villi, we see that they have what I call the Bart Simpson haircut. Let's see if my highlighter will work. Oh, no, it's still not working. But the Bart Simpson haircut here is what we have on top of these cells, and this is called microvilli. And it's basically a little bit of brush border on top of the cells that helps to facilitate and increase the surface area even more. Now, I should point out this brush border doesn't actually move around. It doesn't have cilia on it, but it does increase the surface area for absorption. Okay, so here you can see this brush border under the electron microscope. It is a very microscopic structure on the top of the cell, helps to increase surface area for absorption, but it also contains many membrane-bound enzymes that help to catalyze important enzymatic reactions. For example, we have a sucrase in there that breaks down sucrose into monosaccharides. We also have a lactase embedded in that membrane that helps us to break down lactose. Okay, we've said before that there's 2 different types of digestion going on in the digestive system. There's what we call mechanical digestion, which here is the analogy is our washing machine that's sort of mixing things back and forth. And we also have chemical digestion. That is, we have enzymes and acids and things like that that are working on the food to break it down. Well, most types of digestion are going on in all these different organs. The stomach had the acid and it had pepsin, that's part of the chemicals, and then it also had the agitation and mixing back and forth, which is part of the mechanical digestion. Now, so we had acid, we don't have acid anymore. What happened to the acid? Well, it got neutralized by the bicarbonate that was produced by our pancreas. So we initially had a pH of around 2, now we have a pH back around 6. Okay, so our pH is raised a lot, and that's because the intestines are very delicate, a lot less mucus there, and their purpose is absorption. We do have still a lot of enzymatic activity, but that enzyme activity is provided primarily by the membranes, the brush border of the intestine, and also the pancreatic enzymes. All the enzymes that were secreted in the stomach worked well at a pH of 2, but now that we're at a pH of 5 or 6, those enzymes don't work anymore. Okay, so let's talk about the mechanical digestion that's going on inside of the large intestine. Mechanical digestion in the small intestine. First of all, we have something called segmentation. If you imagine something called chyme, all this food and fluid that's just in a base slurry inside the intestine, which we see up top right, what happens is that it sits there for a while, we absorb some fluid from it, but every few minutes or so, we begin to undergo something called segmentation. So what happens is circular smooth muscle divides that large bolus of food into smaller boluses of food, and then it just sits for a while. So here it is sitting, this food is now sitting in these individual globules that are now sitting in all that soup of enzymes and stuff that's in the small intestine. The fact that we divided this thing up into smaller parts helped us to increase, that's right, surface area. And so the more surface area we have, the easier it is for the digestive enzymes to work on the food, because even though absorption is a job, so is digestion. That's one of the 2 jobs of the small intestine. Okay, so after going through this process of segmentation and then letting it steep in these enzymes, we're going to slap all this food back together again, just like we are at the top of the screen. So we go through this process of cutting all the food up, letting it absorb the enzymes, and then mashing it back together again. And this makes sure that we have a very good enzymatic mixing of the food with the enzymes. Okay, After a while, we're then going to move that food in a particular direction. So the first part here is something called a migrating motility complex. So here we have smooth muscle contraction behind the food and relaxation in front of it. This starts after most of the meal is already absorbed, and now we need to move it to the large intestine so that we get rid of all the waste material. It usually happens somewhere between 90 to 120 minutes to reach the end of the small intestines once it starts at the beginning of the small intestines. However, it does depend on the type of food. As we saw earlier, some foods like carbohydrates are digested very quickly, move through the digestive tract very quickly, whereas fats and complex proteins take a lot longer to move through the GI tract. So here you can see this migrating motility complex as it's pushing food towards the large intestine at the end of our ileocecal valve. Now let's take a look at the histology of the small intestine. Remember, the primary function of the small intestine is absorption, with a secondary function being, that's right, digestion. And so the main cells we have are the Bart-Simpson haircut cells. I call these absorptive cells, and they have the little brush border up top, which is important, again, for not only absorbing things, but also catalyzing enzymatic reactions. Okay. After that, we have something called goblet cells. Goblet cells, if you remember back to 141, secrete goblets and goblets of mucus, and this helps to move things through the digestive tract. And if you eat a lot of high fiber foods, you have more mucus. If you don't eat high fiber foods, a lot less. And then number 3, we have our endoendocrine cells that secrete things like secretin, CCK, and GIP. You should already know what these first two are. CCK, remember, was secreted by the digestive tract, and it helps to regulate gastric emptying. CCK basically puts the brakes on gastric emptying, and it's secreted in times when you have a lot of fatty meal, and some of that fat is reaching the small intestines, and it's basically slowing the rate so that the intestines will have adequate time to break that down and absorb it. And then we have something called Paneth cells that secrete something called lysozyme, and this is an antimicrobial protein that helps to prevent infection of the GI tract. All right, let's talk about chemical digestion of carbohydrates within the small intestine. First of all, the enzymes that are around either came from the pancreas or they came from the small intestine itself. All the enzymes that were working in the stomach are now inactivated because of the change in pH. So we have our pancreatic amylase. Remember, amylase helps to break down amylose, which is a starch. And then we have all these brush border enzymes, which are located in the membranes of those Bart-Simpson haircut cells. And these cells have enzymes such as sucrase to break down simple sugars like table sugar, lactase, which breaks down milk sugar, and maltase, which breaks down another type of disaccharide. We also have protein digestion continuing to go on here in the small intestine. Remember, where did protein digestion start? Well, it started in the stomach under the direction of pepsin. But now we need a different enzyme to digest proteins here since the pH has changed. Okay, so pepsin in the stomach is no longer active. But here we have trypsin, chymotrypsin, and carboxypeptidase and others that come primarily from the pancreas. So the pancreas, the big one, is probably going to be trypsin that really helps to break down proteins. And there's also other enzymes called aminopeptidases that help to remove the terminal amino acids from a protein. Don't really worry about that. The primary protein digesting enzyme you need to be familiar with is trypsin. Of course, we have some other dipeptidases, again, not very important, that split dipeptides into single amino acids. Let's just skip those. Okay, let's talk about digestion of lipids, because lipids haven't been digested up until this point. We didn't digest lipids at all in the stomach, and now this is where we're going to digest lipids in the small intestine. So most lipids are digested in the intestine, and they're digested in a few different ways. First of all, we have our bile, which is there to emulsify the fats. That is, take large globules of fats and break them down into smaller globules, which have, that's right, more surface area. So bile salts do the emulsification, but it's actually the pancreatic lipase, the enzyme, that breaks down those triglycerides into monoglycerides and fatty acids. So that's what you see here. Unfortunately, my fat face is covering up that thing, but here you can see a single monoglyceride and then one or two free fatty acids. So that is the job of our pancreatic lipase. Okay, we also digest nucleic acids there. Remember, nucleic acids are in your cells that help to program them and tell what the cells need to do on a daily basis. They're sort of like the major instruction set. And because you're eating living things, no matter what you eat, one time it was alive. So even if we're talking about a candy bar and the chocolate in there, remember, came from a plant, and that plant had DNA. And so you have enzymes that chop up the DNA of the organisms that you're eating, and then we can reuse some of that DNA, single individual nucleotides and so forth like that. But this is why you do not need to worry about the DNA from your GMO foods ending up in your body. You have enzymes that break that DNA down. You don't have to worry about becoming a genetically modified corn plant just because you eat genetically modified corn. Anyway, here are the enzymes that we have. Our ribonucleases, which come from our pancreas, our deoxyribonucleases, which also come from our pancreas. So ribonucleases break down RNA, and deoxyribonucleases break down DNA. And we have a couple other ones as well. We're not going to go into those. Okay, now let's talk about absorption of nutrients, because remember I said the small intestine is a major organ where absorption of nutrients happens. Okay, 90% of all absorption happens in the small intestine, so it's pretty darn important. Okay, carbohydrates in particular are absorbed amazingly fast. And this is really important because if you're training for a race or something like that, and you want to get a lot of energy in the body and not have it weighing you down the next day, carbohydrates are the way to go because they can be absorbed at a rate of about 120 grams per an hour. This is also one of the reasons why after eating some carbohydrates, you can still feel empty or hungry an hour or two later because they are digested so quickly. Okay, so what happens here? Take a look at the left-hand side. We have glucose or galactose, an example of a simple sugar. It moves into our cells through secondary active transport, or it can move in through facilitated diffusion. And then once it gets into that cell, it's going to move across the brush border and into our capillaries through something called facilitated diffusion. So bottom line here is it's pretty simple. It moves across either by secondary active transport or simple diffusion. And once it's in the on the other side of that that absorptive cell, it's then going to be picked up by our blood capillaries. And once it's picked up by the capillaries, it's going to move towards the liver via our hepatic portal vein. Remember, the hepatic portal vein transports things between the capillary beds in the intestine into the capillary beds in the liver. Okay, let's talk about absorption of proteins. Remember, in order for proteins to be absorbed, they have to be broken down into single amino acids or at the very least dipeptides. So they're absorbed as one or two peptide fragments. Okay. We have to use active transport into the epithelial cell, but once we get into the epithelial cell of the intestine, it then moves into the bloodstream by simple diffusion. Again, it's going to travel to the hepatic portal vein where those proteins, at least the amino acids, will be deaminated, which is an important function of the liver, and that deamination helps to render them safe and makes them non-toxic. Okay, now let's talk about absorption of lipids. Remember that we had our fats being broken down by the lipases in the pancreas. And the lipases help to break down the triglycerides into a monoglyceride plus a couple free fatty acids. But now we have to absorb all that stuff. So the short chain fatty acids diffuse directly into the capillaries, not a big deal. But the long chain fatty acids and monoglycerides have to be transported into the epithelial cells by something called micelles or amphipathic capsules. Now remember, fats are not very soluble in water. And so this is basically a little capsule that we have to put around that fat in order to get it to diffuse into our cells. And let me see if I can draw on here, if it's going to let me draw. Exciting. No, still not letting me draw. Anyway, the point is the micelle helps to move that material into our cell of our small intestine. And once it's across on the other side, it's then going to move into another capsule called chylomicrons. Okay, now chylomicrons are another type of amphipathic capsule that help to transport this lipid through the bloodstream, because once that chopped up lipid gets into the intestinal cell, it gets reassembled into a triglyceride. That triglyceride gets packaged into another capsule called a chylomicron, and then those chylomicrons, ironically, are not going to go by the blood, but they're initially going to catch a ride on the lymphatic system. Again, because these lipids are so very large and they're chylomicron capsules, they cannot be absorbed by the intestinal blood capillaries. They have to be absorbed by special structures called lymphatic capillaries or lacteals. Okay? So the lacteals, the chylomicrons exit the epithelial cells via exocytosis and enter these lymphatic capillaries called lacteals. They then travel throughout the lymphatic vessels until they get to that thoracic duct, which is a place where we can dump the lymphatic fluid back into the bloodstream. And once that happens, that lymphatic fluid gets dumped back into the left subclavian vein, and then it travels eventually to the liver or other parts of the body. Okay. Wherever these chylomicrons end up, oftentimes it's going to be in the liver. The chylomicrons themselves are removed by something called lipoprotein lipase, and it basically pulls those lipids out of the lipoproteins. Okay, remember lacteals we said are those specialized capillaries that we just talked about. They're part of the lymphatic system, and the reason they're important is because our fats, once they're in the chylomicrons, are too large to move through the skinny little tiny blood capillaries, so they have to move through the chylomicrons, or have to move through the lacteals. Now, here's something that's interesting. You've probably gone in some point and given some blood work, and maybe before you go and get the blood work done, they say, oh, you know, don't eat the night before, and you're like, oh, I forgot, I ate this morning, but you still want to go to your appointment anyway, and so you kind of lie and said, oh, I haven't eaten this morning. Well, ironically, if you've eaten a fatty meal, the phlebotomist, once they draw that blood out and spin it down, can actually look at the serum in there and see the little globules of fat that are carried by our chylomicrons. You can see this with the naked eye. It's so disgusting. So I actually was chastised once for eating when I shouldn't have eaten on the morning of giving some blood. Okay, let's talk about absorption of other stuff in the small intestine. That is water, electrolytes, and vitamins. Now remember that electrolytes is just another name for salts, but some of these salts are essential. So 90% of the water that we consume is reabsorbed in the small intestine by simple osmosis. The remainder will be absorbed in the large intestine. And the reason we don't pull it all out in the small intestine is because stuff starts moving more slowly once we pull too much water out. So there's a trade-off between keeping some water in and making the poop go away. And that's what we're interested in. So the electrolytes and salts that are absorbed in the small intestine, again, mostly in there, but we'll have some absorbed in the large intestine. Vitamins A, D, E, and K are absorbed in the small intestine. Again, as long as we have some fat in the diet, that's possible, as well as vitamins B and C, and vitamin B12 or intrinsic factor. That uses active transport, whereas the other vitamins are primarily absorbed through passive transport or just simple diffusion. Okay, welcome back. Now we're moving on to the final part of the GI tract, and that's called the large intestine. The large intestine is large because of its diameter, not because of its length. Its length is only about 5 feet, whereas the small intestine has a length of 10 to 12 feet. Again, it just has a larger diameter. The primary function of the small intestine was absorption of all these nutrients as well as digestion. The primary function of the large intestine is really just absorption of excess water plus some absorption of vitamins and some generation of vitamins by bacterial fermentation. So let's take a look at where food enters. I'm going to try to use my pointer here. And it enters down here at the ileum or ileocecal valve. That's where food moves into the first part of the large intestine, and that part right there is called the cecum. It then moves up from the cecum up to the ascending colon. It goes across from the transverse colon, and then down the descending colon, and then finally sigmoid colon, rectum, and anus. And this can take a few hours depending on the type of food that we've eaten. Okay, so let's take a look at some anatomy of the large intestine. The first thing you'll notice is that the large intestine is divided in these pouches called haustra. So here is a haustra right here. It's a big pouch. And these pouches are basically where the food will sit while it dries out. The primary function of the large intestine is absorbing that last 10% of the water. And as it does that, the pouch will just sit in there and it will desiccate, and eventually it'll move to the next pouch. We then have something called Taenia coli and epiloic appendages. These are these structures right here. The epiloic appendages are just these little sort of globules of fat right here. They're not very important. Okay, now let's take a look at the internal anatomy of the large intestine. Now, like most of the parts of the GI tract we've talked about so far, it's made up of simple epithelium. And remember, simple epithelium is important because it facilitates really good movement of things across the membrane. And so here we're still going to be absorbing some nutrients, that is vitamins and minerals and things like that. And also we're going to be absorbing some water. So we have two different types of cells that are important here. First is our absorptive cell, and our absorptive cell, if you remember, has that Bart Simpson haircut. There it is. And that is the microvilli that helps to facilitate movement, in this case of water, across the membrane from the lumen of the large intestine into the actual blood capillaries. We also have our mucus cells, and these mucus or goblet cells secrete lots of mucus, and that's particularly important in the large intestine because mucus is what keeps things going round. And probably being little youngsters and things like that, you don't appreciate the fact of having regular bowel movements, but believe me, your patients will as they get older, because having enough mucus in the intestine is important to making sure that people are regular. All right, let's talk about the functions of the large intestine. I really already hit on them already. We talked about it that its major function there is to absorb excess water, and that's true. But we also have some other functions that are going on here as well. One is the haustral churning that we talk about, this kind of back and forth movement of stuff going from one haustra to the next. Actually, that's just one direction. It doesn't go back and forth. And then we have something called mass peristalsis, which is pretty funny, and I'll talk about that here in a minute. In the large intestine, we have bacterial production of vitamins B and K, and that happens through bacteria. We can't extract these vitamins ourselves, and so that's why it's important to have a good gut flora of bacteria. And also the large intestines is where we absorb remaining electrolytes and water, and eventually we get rid of all the other stuff we don't need through the act of defecation. Okay, haustral churning. I alluded to this earlier. We said food goes into one pouch. It sits there for a while. As that food sits there, we continue to absorb water from that food, and then bloop, it gets squeezed from that pouch to the next pouch. It sits in that pouch for a while. It absorbs a little bit more water. Bloop, moves to the next pouch. Sits there for a while, absorbs more water. Bloop, goes to the next pouch. Okay, you can see what's happening, that as we go, let's say, up the ascending colon and across the transverse colon, the food, or in this case just fecal matter, is drying out, and that's important because large intestine major function is to reclaim some water. But when we get about midway across our large intestine in our transverse colon, something happens called mass peristalsis. And you have all felt this before. You're sitting in a meeting or on a date somewhere, and all of a sudden your intestines go and you're like, whoa, and you have to clench your butt cheeks so the poop don't come out. And so that happens as food moves rapidly across the transverse colon, down the descending colon, down the sigmoid colon, into the rectum. And the only thing between you and calamity is a little bitty strip of skeletal muscle down here in your anal sphincter. Otherwise, that poop would just come shooting right back out, and you would be embarrassed as hell. So this is an important reflex that helps to move the fecal matter across the transverse colon into the rectum once the majority of the water is absorbed. It can be initiated by something called the gastrocelic reflex. So if you've just eaten a big meal, basically there's endocrine and nervous communication between the stomach and the large intestine, and it's a way of telling the large intestine, hey, food's coming, it's on the way, you need to get rid of this stuff so we can have more room for the new food coming in. So that's sometimes why you have to poo after you just ate. Okay, so chemical digestion and absorption. There aren't any enzymes secreted in the large intestine. That being said, we do have lots of bacterial there that have enzymes of their own that are really helpful in breaking down some stuff that we otherwise could not break down. So in the process, we are producing some wonderful intestinal gas, which consists of carbon dioxide, methane, and hydrogen, as well as some hydrogen sulfide, which, if you don't know, smells like swamp gas. Now, during the transit of the large intestine, the, the bile that was moving through our large intestine has now been converted into stercobilin. So it goes from a green coloration into a brown coloration. And then vitamins B and K are produced by bacteria and absorbed by the simple epithelia of the intestines. And so the excess water, the remaining 10% that we can get out, is pulled out. Now, is feces completely dry? No, it's actually still fairly moist in most cases, but we get as much water out as we can. Okay, let's talk about feces formation and the defecation reflex. Now, remember that feces dries out as it transits the colon. It goes from one haustral to the next, and as it does, some of that water is absorbed. That mass peristalsis we talked about starts about midway across the transverse colon and then forces that food into the sigmoid colon and rectum. And once the rectum is distended sufficiently by this fecal matter that's accumulating, we send nerve impulses to the central nervous system by afferent sensory neurons, and the CNS sends a message back via parasympathetic neurons to the rectum that says, hey, go ahead and poop. But sometimes, you're not ready for that. Remember that the defecation reflex is actually initiated by smooth muscle, so it can actually kind of start on its own. But the thinking part of your brain says, no, I can't just poop here in my chair. So the skeletal muscle that's in the rectum will prevent that. Okay, so the longitudinal smooth rectal muscles contract, shortening the rectal pouch. These are smooth muscles, meaning this happens automatically without your consent. The inner anal sphincter, which is smooth muscle, also opens because it's involuntary. And again, the only thing between you and calamity is this little bitty external anal sphincter that is composed of smooth muscle. Let's see if I can draw on there. So here's the external anal sphincter right there. And so that is what is keeping the poop in. Okay, now once you decide to defecate, that external anal sphincter opens up and then the feces can come out. Now this is an important point to part out with potty training in kids, is that, ideally people are trying to get their kids potty trained as early as possible. I have a 1-year-old and I would love for her to be potty trained. But there is a point in development in which we don't even have possible nervous control over the skeletal muscle in the external anal sphincter. So there's a point at which it's just not even makes sense to try to potty train your kids because at that age they just can't control the reflex.